Good morning, afternoon, or evening to you, wherever you are. I'm Arsalan Arif with Endpoints News, and our topic today is a new platform for target to hit, from fragment screen to structure-based drug discovery. We're sponsored by Wuxi AppTech, and it's my pleasure to introduce our expert today, Dr. Nuska Tschammer. Nuska is the head of Dell Lab Operations Europe at Wuxi AppTech's HITS Crelux in Munich. She's an expert in the field of early drug discovery, previously serving as the head of research and development biochemistry at Nanotemper Technologies. And she's a winner of the Innovation Award in Medicinal Chemistry, which is jointly awarded by the Society of German Chemists and the German Pharmaceutical Society. She has an approximately 30 to 35-minute presentation, and then I will ask Misha questions from the audience. You should hit that Q&A button to get them in live, and if you're watching on demand later, someone will follow up with you by email. And now, without further ado, I give you Dr. Nuska Tschammer.
Thank you, Arslan. I am really happy to have an opportunity to present you a new platform for target to hit. So some of you might already heard that Wuxi AppTech has a slogan, every drug can be made and every disease can be treated. So our ultimate goal is to enable anyone and any company to discover and develop pharmaceuticals and healthcare products to benefit patients. And of course, to achieve this goal, we continuously strengthening our capacities and technologies, and our HIT-S integrated unit is committed to provide excellent target-to-hit solution to all our partners. What that means in detail. So in this presentation, I will briefly talk about modern structure-based drug discovery, then cover some fragment screening principles, and at the end, I will talk briefly about how these guidelines are implemented to our new target-to-hit platform at Wuxi AppTech, which is called HIT-S. And there's nothing better than to practically a case study to present what I just practically theoretically covered before. So now I would like to go a little bit back in the past and remind you on the thalidomide story. I think that I don't have really to lose a lot of words on this story, where practically about 10,000 infants worldwide were born with different birth defects. And at that time, of course, thalidomide was broadly prescribed as a drug against morning sickness for pregnant women, and it was completely claimed to be completely safe. But unfortunately, our body is a black box for in some cases, or at least at that time, and it was really a question why suddenly these strange phenomena were observed. And it took some decades before we actually were so advanced in our innovations in structural biology to be able to identified mode of action. And as shown on this slide, practically, it's not all practically the structural biology per se, that means X-ray crystallography as the first approach in structural biology, actually does not suffice to practically tell the entire story. Important part of structural biology are also adjacent sciences, and it's really interwoven network of biochemistry and biophysics. As shown here, first, of course, before we actually come down to structural biology, we have to start with very good, let's say, reagent, in this case, recombinant protein. And of course, to do that, first we have to apply methods of molecular biology to clone the cDNA and to express protein in the host system, and then to get it out in very pure form. Then we can move with this protein in biophysics studies, where we estimate how our compounds of interest, in this case, thalidomide and some related substances, interact with the protein. And then, if everything works great and we have a very, let's say, strong evidence that the compounds interact with the protein, we go down to structural biology, in which still protein X-ray crystallography is the method most commonly used and also the method with the highest throughput and the highest resolution provided. And in combination with all these approaches, we now know exactly why practically thalidomide causes these severe birth defects. At the same time, actually, structural biology helped to identify mechanism of action, and on the other hand, gave the birth to so-called immunomodulatory drugs. That means we discovered what this black box is, and now this is not a black box anymore. We know exactly what happens with the target, and this is a protein cerebron, DDBY complex, and practically the binding of thalidomide to cerebron changes its preferences for the substrate, which is then deemed to be destabilized, destabilized and destroyed, or excuse me, marked for the destruction in the cell. So this is actually nothing new. That's practically our bodies or nature uses small molecules to practically enhance or mitigate the effects of different proteins in the cells. So by nature, in nature, this is, for example, no, I forgot it. So sorry. Auxin is actually inspired by nature, and auxin, you know, is actually the protein which helps plants to go in the direction of light upfront. So this protein, this small molecule acts as natural protein degraders. So next to these protein degraders, and based on the telomerase story, we actually came out with an entire class of drugs, including proteolysis targeting chimeras. And as shown in this slide, again, we have a nice interwoven network of biophysics and structural biology to elucidate this function. So why, again, we need practically other methods to support structural biology? So here, of course, we have a very nice resolution at almost atomic level, how different proteins interact, how different subunits interact, and where our proteolysis targeting chimera binds. Still, this does not tell us what that means practically when the proteins are, let's say, free in solution, when they freely move, because of course this crystal structure is only a snapshot in the time. So when we perform the analysis like shown here using microscale thermophoresis, about which I will talk a little bit later, we see that practically the affinity of this CC885 for the binary complex. That means only when we have together cerebron and DDB1 is actually, average, let's say 17 nanomolar. As soon we add the binding partner GSPT1, the affinity of this CC885 increases a lot, practically up to 1.8 nanomolar, which means we experience here very strong positive cooperativity, which is very important for the function of proteolysis targeting chimeras. So I have spoken about interwoven state-of-the-art biophysical methods, and here also another part which comes into play. These are computational networks, computational methods. And practically with the combination of biochemistry, structural biology, biophysics, and of course, state-of-the-art computational platform, structure-based drug discovery enables target to hit and hit to lead campaigns. Here, practically, you can see that now when we have practically human genome sequence, we are practically investing a lot of time and effort to practically try to extract and purify the proteins which we identify. And of course, the most of them have to be closely characterized and identified as potential targets for the drug discovery. And once we actually are able to produce and extract highly pure protein, we can move on with, so to say, classical part of structural biology. So first is X-ray crystallography, where we have to first design the protein, which is able to crystallize in a yield suitable diffraction pattern, and then we can perform investigation of electron density and assignment of the structure. So if we are lucky, we get a very nice 3D structure. But in some proteins, as you probably know, they can be really tough to get them crystallized or not to crystallize at all. So we need, of course, some other method. So NMR is an excellent approach for smaller proteins, let's say smaller than 40 kilodaltons. Nice about NMR is that you practically don't need to modify the protein. You don't need to perform mutations. You are not labeling the protein, except of course for isotopes. And then you assign the structure. But of course, assignment of the structure in NMR experiments is much more tedious than in, for example, in X-ray crystallography. In last year, cryo-EM is taking more and more significant space in the structural biology. Still, there are quite some limitations which we have to be aware of. And one of them is, of course, this is the size of protein. So in this case, not the protein should not be small as possible, but in this case, the protein should be as large as possible. The other thing is quite nice about cryo that we need quite less material, but still, I mean, always is good to combine as many methods as possible. So when we once have practically the structures, then is the question, so what we do next? So one option is that we actually continue with, so to say, computationally assisted methods to discover active compounds. So we can screen over various databases, virtual databases, where we, of course, start with the structures which we obtained before, and we can use docking and some virtual screening. Then, of course, we can choose compounds, but still we have to synthesize them, we have to test them using biophysical and biochemical method, and then continue in the next level when we have one lead compound. But here, in this case, we can go also other directions. So we can say, okay, we have protein here, but we would prefer first to start with real compounds. And in that case, we can use this protein for screenings like fragment screen and go then practically already with the known fragment in structural biology part of the project. Or we can say, let's use DNA encoded library and start with more lead-like or more drug-like molecule, and then we repeat the same. So no matter what actually our starting point is, the modern structure-based drug discovery is always interwoven network of biophysical, computational, and biochemical methods. So still, I mean, very often, structural biology is discussed only as a tool to enable design and optimization of chemical structures with the goal to identify a compound which is finally suitable for clinical testing. So more or less, yeah, acceleration of medicinal chemistry campaign. But of course, structural biology is much more. We can use it for target identification and validation. We can use it for assessment of ligandability and druggability, and of course, for hit identification, like for example, in fragment screening. So, especially fragment-based drug discovery and structural biology methods are really, as some say, match made in heaven. So why? With fragment-based drug discovery, with nice library of fragments, you really cover vast chemical space by just using a few thousand fragments. So that means your library is actually small compared to HDS library. And that means that at the end also your costs for the screenings are less. And what are the advantages of Fragment? Because I mean, you will say, okay, Fragment is simply a compound which has about 250 kilodaltons or less. So there's nothing, this is nothing drug-like, there's nothing lead-like yet in this stage. Yes, that might be true. And of course, you have to do quite some of medicinal chemistry effort, but here's actually where the methods of structural biology will offer you all the tools which you need to practically proceed down practically your path in medicinal chemistry optimization as fast as possible. An advantage of fragments are that actually while they are so small, they can identify unique binding pockets. And they can also bind small pockets which are available on the protein surface, like protein-protein interactions, which are, of course, highly relevant when you try to discover some new protein degraders or something similar. We can efficiently sample chemical space. So with, let's say, 1,000 fragments, you can, chemically speaking, you're sampling nearly trillions of compounds, because you are focusing on the core, on the practically on preferred structures on something very special. And advantages, of course, I mean, when you go into the screening where your practically compound is already more, let's say, drug-like, sometimes you have to do a reverse engineering because the pharmacokinetics profile is not suitable for your applications. With fragments, you have ability to optimize pharmacokinetic profiles simultaneously with the potency as fragment to hit growth. So that means you have less reverse engineering. And of course, the key enabling technologies are structural biology methods like protein X-ray crystallography, NMR, and then of course, if you have structure, then you can do docking studies and you can do fragment growth in silico. So which methods are most commonly used for fragment screening? So for fragment screening, we need high sensitivity biophysical methods to detect the binding of this small fragment to the protein. And this is no surprise that practically fragments have usually very big affinity, 100 micromolar, even to 10 millimolar, depending on the library. And this, you know, you really need some highly sensitive biophysical methods to detect this binding. So it lies on the hand that you need state-of-the-art biophysical methods like SPR, X-ray crystallography, NMR, or microscale thermophoresis, or new on the market is actually a device, the Entus, which I will introduce later. And practically key component of nearly all fragment screening campaign is actually structure-based drug design. At this point, you might actually ask, so what if we fail to obtain the structure because the protein does not want to crystallize, is too small for cryo-EM, and with NMR, and for NMR is actually a little bit too big. So if everything fails, then of course you can do also so classical, practically medicinal chemistry guided fragment to lead development. And this still nowadays happens in about 30% of all cases. As I already mentioned, people really prefer certain methods to use to detect the binding of the fragment. Ligand observed NMR appears very often. Then you can perform fragment screen using X-ray or even protein observed NMR. So let us compare side to side what these methods can do for you and actually how they compare, what is the advantages and limitations. NMR, of course, if you decide, you can observe either ligand or you can observe the protein. If you decide to observe protein, then you have to label the protein with isotopes so that you can then assign the signal to practically amino acids accordingly. If you observe ligand, then of course you don't need to label your protein, and also the consumption of the protein is much less. You can screen a couple of thousands compounds at the same time, fragments at the same time in the cocktail. Advantage of the NMR is that it can derive structural information, of course, when you decide to use protein observed NMR. And limitations, yeah, large library screenings are challenging, and as I mentioned before, you might need large amounts of isotopically labeled protein if you want to screen thousands of compounds. So usually the people decide practically to do this only with small subset of fragments. X-ray crystallography, on the other hand, is a method that uses protein's crystal structure to determine the binding mode of fragment. It can screen few hundred to 1,000 fragments. It is a huge advantage of this method because at the same time, when you get the confirmation of binding, you also get the structural information at atomic resolution. So it means you know exactly where the fragment binds. And this enables you the very fine mapping of fragment binding site. Of course, the limitation is that you obtain no affinity information, and X-ray crystallography requires relatively large quantities of protein, which of course has to have an intrinsic tendency to crystallize. So what are the options if we say, okay, I don't want to use structural biology for a fragment screen? So next you can go on the next step, or the most commonly used methods are then other biophysical methods as surface plasma resonance and microscale thermophoresis. In this case, you might wonder, I mean, biochemical assays, they are practically marked here, and you would say 50% of all cases, fragment screen, at least in this publication, they were done by biochemical assay. Yeah, that can be done. And practically in this publication, the proteins which were analyzed in biochemical assays were kinases. If your protein has no obvious enzymatic activity, of course, it will be not possible to perform biochemical assay. Also, biochemical assays are known to have a rather large portion of false negatives because, I mean, remember, you have a quite very poor affinity of the fragment to the protein target, and then practically this binding event has at the same time to influence the function of the protein. So, I mean, there is a high chance that you will come out with very small number of hits, if any. So, on the other hand, practically, biophysical assays as SPR and microscale thermophoresis, they actually detect only binding event. For surface plasma resonance, you have to immobilize the protein on the surface. On the optical biosensors, you can screen a few thousand molecules at the same time. Advantages, you have high sensitivity and you get accurate affinity measurements, and of course kinetics. And the protein consumption is also much lower compared to NMR or X-ray crystallography. Of course, every time when you modify the protein, that might change the conformation of the protein and cause some inactivation or even low stability. Microscale thermophoresis, on the other hand, is a technology which where the measurements are performed in solution. Similar to SPR, you can screen a few thousand molecules, and the advantages are similar. It's very sensitive, and of course, in solution method comes with a lot of advantages. Limitations, although that you can use intrinsic protein fluorescence to perform this method, most of the time fluorophore labeling is required. But then you can say, okay, that means I have to modify the protein in the same way that practically almost for surface plasma resonance. Still, we observe that practically the proteins behave on the surface completely differently than in solution, even if they are labeled. So I have told you a story now, brief overview, provided you a brief overview of our modern structure-based drug discovery, interwoven network from protein production over screenings, let's say fragment screening, I focus on the fragment screening, structural biology, and then biophysics. And all this entire concept is practically implemented at WuXi EPTEC HIT-S unit, which is a new target to hit platform. We are located in US, Germany, Munich, and in China. And this is actually interwoven network of 4 locations, as you can see here. And no matter where you decide with which unit you decide to work with, we work as one. And no matter what you do with us, we will start with protein, we offer screenings, we have a state-of-the-art biophysical platform, and we cover all the methods of structural biology. So why do we start with the protein? So as Frances Arnold, Nobel laureate in chemistry, said, you get what you screen for. So some of my colleagues, they like to say garbage in, garbage out, which is actually at the end really true. The fact is the protein is the most important reagent in this system. And the quality of reagent will determine how successful your screening campaign will be. So we really pledge to deliver the protein of the highest possible quality. Very often we say that's nearly crystallographic grade protein. So we always aim to reach over 90% purity. The protein which is not aggregated, it's correctly folded. And we use various biophysical methods to determine if the protein is really properly folded. It's like we use, for example, binding to positive control if there is any, in, for example, the forming of expected protein complexes. Then you can do screening with us. As I mentioned, either fragment screening or, for example, DNA encoded library screenings, and then we go down the path with you. If we start with fragment screen, then of course, next stop is a structural biology platform. As I mentioned, we cover all 3 platforms, and we still X-ray crystallography is our main working horse. It's simply practically the throughput at X-ray crystallography is unprecedented, and also the resolution of the binding site, which you can obtain with X-ray crystallography, is also not easy to achieve with other methods. Then we offer NMR, also, for example, fragment screen by NMR, and also cryo-EM with our close collaborator AN, which is the company which is located in Czech Republic, in Prague. So now I would like to describe you, so that you had a better feeling how we do this, I would like to introduce you to a case study on STING. This is stimulator of interferon genes, which is actually facilitator of innate immune signaling. It actually recognizes cytosolic DNA from bacteria and viruses, and forms a dimer and promotes the production of the type 1 interferon, ENF alpha and EFF beta. The idea behind is that practically, if we find the activator of STING, this could be utilized as a therapeutic for cancer and infectious diseases, because you want to stimulate the cell to elicit immune response. And of course, in autoimmune diseases, our immune system is a little bit too active, so then you would need inhibitor of STING to practically suppress this undesired high activity. So typical workflow at HIT-S actually mimics well-established approaches. Here, what I would like to talk today is so-called TRIC primary screen. This is a method similar to MST, where we identify binders at one single concentration. In the next step, we use up to 8 to 12 concentration to determine the hit, to validate the hit and obtain the KD. And then we use a different orthogonal method here. In this case, we use 3, MST, SPR, and nano-DSF to confirm the hits. And then we moved on to X-ray crystallography with fragment soaking and STD NMR. So what is TRIC? So this is actually very new technology on the market, which is actually well characterized and well known. So it actually identifies or quantifies molecular interactions by observing the change in fluorescence upon increasing temperature. Because of that reason, you need first your target molecule labeled with the fluorescent dye. Then we applied very precise and brief laser-induced temperature change, and we observe how the fluorescence diminishes with increasing temperature. So what influences these changes in the fluorescence? So you might have heard of microscale thermophoresis. So in that case, practically there's a movement of labeled molecules away or practically in the temperature gradient away from the source of heat, so to say, and this will actually increase the impact the change in fluorescence. But in the case of TRIC, so-called temperature-related intensity change, this signal is affected by fluorophore microenvironment with changes to the binding event. So, and this means that actually either ligand is bound very close to the fluorophore and actually either enhances or quenches the fluorophore, or we have to do with the conformational change, and this microenvironment changes by ligand proximity, and this influences the fluorescence signal. Basically, we label our mixture, we label our protein, we add the ligand, we heat up the solution with infrared laser, and then we observe the signal in time. Unbound state, in unbound state, the protein, the fluorescence of the target will change differently than in the bound state, and this difference is so-called TRIC signal amplitude. And this can be then plotted and translated into KD. So we use practically this method to perform our case study. So why we decided to use TRIC? So because this is really very fast. For example, for the validation or assay development and validation, we need 1 week. We are able to specifically label the protein only on the his stack by using so-called red tris NTA dye, which specifically binds only to the his stack of the protein. That means the rest of the protein is intact, and we don't have to worry about the conformational changes of the protein caused by labeling. This measurement takes place in 348 well plate, as shown here. And as you see, for about 2,200 fragments in duplicates, we needed only 8 hours of measurement time to perform primary screening. So that means you start your assay, let's say, in the evening, and you come in the morning, and you already, and this pie chart waits on your screen. So, you know, this is very nice, very fast. So this is how the machine looks like. And of course, in next step, we have to perform hit validation. This also went very fast. We actually decided to screen or to determine the KD for 162 primary hits in KD response, and we confirmed 80 binders. And as you can see, you can determine 348 data points within 30 minutes. So then practically you can see that with this method, your fragment screen is done more or less within a week or 2. Then, of course, I mean, After first round of primary screen, then in a hit fragment hit validation, then you have to confirm your hits by other orthogonal methods. We have decided then to use MST. We use the same labeling strategy. Then, of course, different method. This is SPR, where we use immobilization of biotinylated sting. And at the end, we decided also to see if fragments induce some thermal stabilization of the protein. We use nano-DSF. In this method, it's actually very nice because you observe only intrinsic tryptophan fluorescence of the protein. So you're adding no dye, and you don't have to worry that practically this dye might interfere with the binding of your fragment to the protein. At the end, practically, we confirmed 66 binders, which they have actually very nice affinity, practically high affinity for the fragments between 10 and 500 micromolar. Then, of course, we went down the stream, and as I mentioned, next step is structural biology. We decided actually to try to co-crystal, not to co-crystal, we wanted actually to confirm the binding of fragments by soaking. So in parallel to the screening campaign, we already practically produced crystal-grade protein and established soaking system, which we soaked with fragments in cyclic D-GMP as a control. And after automated analysis, followed by manual refinement, we were actually a little bit surprised that we actually observed only cyclic D-GMP bind, but practically with fragments, we have observed no fragment bound. So now you might say, okay, so this is strange. I mean, you have fragments, they're supposed to have high affinity. Why they don't bind to the to your protein of interest? So, I mean, don't forget, practically crystallography, there are crystals. So it could be that fragment binds somewhere here on the surface, which is actually, when you have crystal, is not accessible. Yeah, or the protein is in the conformation, which is again, does not allow fragment bind. So we said, okay, soaking, not really a good way to go with sting. So currently we are co-crystallizing fragments. Of course, if you are co-crystallizing, then you can induce different crystal structures. The fragment is there when the protein creates crystals. So you have really much higher chance that fragments will be trapped in the crystal or bound to the protein. Still, we said, okay, we need structural confirmation. We want to have really a validation of the binding by structural biology methods. So we said, okay, next best is, of course, NMR. And we decided for saturation transfer difference NMR, which is very simple and fast method for ligand screening. And this is also the way how you can perform fragment screening. Here is shown what practically happens here. The exchange between free and bound ligand allows intermolecular transfer of magnetization from the receptor, like shown here, to the bound small molecule. And you can see here, because practically the fragments are kind of binding and unbinding, this saturation transfers to the ligand. And this is what you can observe. And when you subtract the spectra, you can see practically, you can see the ligand. So yeah, of course, first you have to set up the experiment, and our cyclic D-GMP was, of course, again, used as a positive control. As you can see here, we see a very nice binding. Clearly, this is our positive control, and where we have no protein in the mixture, we observe no signal. So we decided actually to test 11 best fragments, and we obtained 6 confirmed binders, 3 potential binders, and 2 appear to be as non-binder in NMR. And here you can see example of the analysis for the binder, potential binder, and non-binder. So Bottom line is this modern structure-based drug discovery is actually a very interwoven network which benefits largely a lot from biochemistry, biophysical methods, computational methods, and really enables target to hit and hit to lead campaign. And these processes are carefully mimicked by the WuXi Aptec's HIT-S platform. The core of this HIT-S platform is fragment screen, which is suitable for challenging targets, efficient sampling of chemical space, and identification of unique binding sites. To get the best out of your screening campaigns, we provide high quality proteins for screening, structural biology methods, biophysics, or biochemistry. And in our biophysical department, we have several biophysical approaches accessible. We are proud to be a certified provider of microscale thermophoresis and MST and temperature-related intensity change technology, which is hidden in the device, the Antus, because these are really very innovative approaches for fragment-based drug discovery and always prove their unprecedented sensitivity for the fragment. And again, WuXi Aptec HIT's platform provides really a one-stop solution for clients seeking support for this target to hit phase. Thank you very much for your attention, and I'm open for questions.
All right, thank you very much, Dr. Tschammer. I'd like to remind the audience to get your questions in right now. Use that Q&A button down there. I've received several, but it is not too late to ask more. Okay, let's get to the first question, Dr. Tschammer. I can see that more biophysical assays would give better understanding of binders, but what would be the minimum number of biophysical assays that you recommend?
So so we usually recommend 2 to 3 biophysical orthogonal biophysical methods to confirm the hit. This is standard. What we actually have noticed that, of course, all the methods have their advantages and limitations as presented, and we really want to make sure that we are not missing a nice hit.
Okay. All right, a couple more here for just a little bit of time that we have left. Okay, in FBDD, in fragment-based drug discovery, when determining whether this is a valid hit to move on to a clinical drug target design, is it mandatory to have a structure, X-ray, or NMR?
No, it's actually not mandatory. So, I mean, if there is a chance to obtain the structural information, I would absolutely go for it, because then you're not tapping in the dark. So medicinal chemists, they really love structures. They really love to combine them with computational methods to optimize the, so to say, fragment to let's say more lead-like or drug-like molecule. In the case that this does not work, then you really have to go practically the classical medicinal chemistry way, as they did it 30 years ago, where you simply synthesize a lot of different analogs analogs actually, and then you practically move slowly, and then you go back to screening, to characterization, and then you go on. So you always have to do these iteration cycles. But even if you have no structural information on the protein, you still can successfully finish the fragment-based drug discovery campaign. And according to the literature, this is about in the 30% of all cases. But so far in our hands, until now, we were able to obtain structural information for all screening campaigns.
All right, wonderful. Okay, just time for a couple more questions here. Is there any difference or overlap in the affinity range between DEL and FS? Do they both cover PM to low MM?
So this is interesting question. So of course, don't forget that practically for when you are using DNA encoded libraries, you're starting with much more drug-like molecules, which practically means that you can even find the nanomolar binders, and of course up to millimolar binders. Everything is there. In fragments, of course, fragments are very small, very limited in the size, so you can expect weak binders in micromolar to millimolar range. So partially they overlap, but of course, if you search immediately for hits with a higher affinity, then you have to choose, let's say, DNA encoded libraries or similar.
Okay. Here I have another question from the audience, Dr. Tschammer. Do you have in-house chemical libraries of fragments or DNA encoded libraries, or are they provided by the customer for the individual screening project?
So we have actually our own in-house fragment library, and of course our own DEL libraries. We are actually quite proud, very proud of these libraries because they really incorporate the know-how of chemists at WuXi, because you know WuXi is it's a huge chemical company. So we have quite some unique scaffolds which are hidden in this library. However, if the client says, okay, I would like to have my library screened at your place, or I would like to have a focused library produced around specifically for my target, this is not a problem. We are more than happy to provide this type of services too.
Got it. Okay. Another question here. Since protein quality is crucial, what if there's no commercially available tool compound? How would you then assess the protein quality?
So this is a tough one, but actually this is actually the situation which we are occurring very often because people are interested in completely new targets. So what we actually we go with biophysical methods to practically with the intention to confirm the appropriate fold of proteins. So for example, nano-DSF would be one of the methods to determine if the protein is properly folded. Then we can do also CD or something else. So yeah, we have to practically indirectly then confirm that the protein is properly folded. Very often we then know practically that this protein has a certain binding partner, and then of course we can see if we are able to measure or if this complex is formed. So there are ways to go.
Okay. All right. A couple more questions here. Let's try to squeeze in. Briefly, could you tell us what are the time frames for each step of the process?
So fragment screen, depending on the method which you're using, it can take between, let's say, 2 to 4 weeks for the primary screen. Then, of course, you have to choose some orthogonal methods. Depending on the orthogonal methods, you have then additional week or two. On top of that, it means you have a list, your bucket list of fragment hits within 6 weeks. Usually, we start crystallography experiments in the parallel so that we have crystals ready or system ready before you are done with the screening. So it's usually in about the same time frame. So this would be, as I mentioned, this is about 6 weeks and then maybe some additional 6 weeks for the crystallography effort. So if everything goes well, you have fragment-based or you have fragment screen, including structural information available within 2 to 3 months. With their screening, we need about 1 month. So there you are practically in 1 month from the hit to your list of confirmed hits, or at least of hits, so to say. And then we continue with the off-DNA synthesis, which might take a couple more weeks, but still, no matter which screening you go, your move or the time frames are between 2 to 3 months.
Okay. All right. And I think I've got one last question to finish our panel today. So I'm going to read this to you. My question would be about the experiences with the screening method. MST is very rare according to your slide. Why is MST not used more often? And how do you decide between SPR and MST?
Yeah, so MST is actually now on the market for, let's say, about 10 years, a little bit more than 10 years. And of course, you might feel that it's not easy for a new method to get established. But in the last years, we see more and more successful screening campaigns, which were fragment screening campaigns, for example, or other campaigns which were performed using MST. So the tendency is increasing. In our hands, we actually noticed that practically you can simply not screen certain targets using SPR due to various reasons, and these targets are still well accessible by MST. So how to decide MST or SPR? I mean, for that we have a very simple question. We start assay development for your protein of interest on both methods simultaneously. And then the method which yields the best, let's say, sensitivity, assay window, robustness, then we continue with that method. Sometimes might be SPR, or it can be TRIC, or it can be MST. Yeah. So we let the protein to choose.
Okay, all right. And with that, we'll conclude today's webinar. And once again, thank you to our sponsor, Wuxi AppTech, and Dr. Nuska Tschammer for sharing your expertise with us today. For Endpoints News, I'm Arslan Arif, and thank you for joining us, and we will see you next time.